Hello, welcome to scQuery, a web server for comparative analysis of single-cell RNA-seq data. Let's go through a quick tutorial on how to use the site. On the home page, you can see an embedding of a selection of our data split up into a plot with anatomical features and another with more specific cell types. To query with gene expression, you can upload your RPKM data here. Note, however, that you should use our pre-processing pipeline to produce this RPKM data file. Our pipeline is linked below with documentation. The server only accepts data produced by this pipeline. Or you can jump right in with a pre-made sample query of a selection of brain cell types. So let's do that. We first land on the Similarity Heat Map tab, where each row is one of the cells in our query file, and each column is a cell type in the scQuery database. Both your uploaded sample query and our database are transformed into a reduced dimension space via a neural network. Then the similarity of each query cell to each database cell type is computed in this reduced dimension using nearest neighbors. And that's what this heat map is showing, where darker green means more similar. Rows are clustered and can be explored using the branches in the dendrogram on the left. Please take note that none of the other tabs are active or clickable when you first upload the data. Each of these tabs do their analysis based on a cluster, and thus you must first select a cluster before you can use them, and so I'll do that now. Now the other tabs are activated. Let's quickly go through what each tab shows. The Cell Types tab shows how similar your cluster of cells are to a particular cell type on a scale of 0 to 1. You can click the Cell Types button and scroll down to find the cell type of interest. You can use the slider to select a cutoff on the similarity score. And below, you can see a set of your sample cells with their similarities. You can also click on the differential expression heat map button . This will open up an image showing the differential expression of genes as rows and cells as columns, where the shading is based on the log average RPKM of those genes. These genes are the top up and down regulated genes we've identified specifically for this cell type. The rightmost column is the log average RPKM of this cell type in our database. The Nearest Neighbors tab will give you an idea of the distribution of cell types in your selected cluster. Each bar represents the total number of times that a particular cell type appears as the nearest neighbor for a cell among all of the cells in your selected cluster. The Low-Dimensional Mapping tab shows a visualization of your query files in a reduced dimension. You can view cells from the scQuery database alongside your query cells by clicking on cell types in the legend. You can click and drag to zoom in and explore the data further. In the Ontology Graph tab, a subgraph of the cell ontology which contains the cell types that are hits for your selected cluster are shown. If I click on one of the green nodes, such as cortex, then the Differential Expression tab is now activated. There again, we see the log average RPKM for the top up and down regulated genes that we have identified for this specific cell type. The leftmost column is the log average RPKM of these genes among all the cortex cells in the scQuery database, and the other columns are cells, individual cells in your selected cluster. Double-clicking on any column label will sort by that label. Finally, we have the Metadata tab. In this tab, we can see some metadata about the studies that are contained within the set of cells that are hits for your query cell. The similarity score for a study is the sum of similarity scores of the cells within for the particular query cells that you uploaded. You can see things such as the PubMed link, an overall design, summary, and more. As an alternative to querying by expression, you can also query with a list of genes of interest. Your query list is compared to a set of cell type specific gene lists that we have identified through differential expression analysis, and you can explore which cell types are most enriched for your query list of genes. We also provide an example query for this. Also, at the top, you can see links for our publications and any supplementary files referenced in these publications. There's also a link to download all of our processed data, including the metadata and our labels. Lastly, there's a forum where you can make posts to ask for help or give any feedback, which we would greatly appreciate. That's it for scQuery. Thanks for watching, and we hope you find it useful.